maybe a little bit before you were born. Did somebody just try to join? Okay. Oh, good. You're recording. Okay.
Oh, yeah, Mason, this is me. And I just started.
And so the SARS outbreak happened a little bit before you were born, in 2002, and it was by bats, and it spread to Hong Kong and caused a global outbreak. If you know, there's a lot of news going on about on the time. The U.S. wasn't particularly affected by the SARS outbreak, and that's one of the reasons why generally people say that we weren't as prepared for the coronavirus is because we weren't actually hit by the SARS virus, which was less extreme than the coronavirus. In the sense of its rate of infectivity and how many people it has infected, SARS is significantly smaller. But because we weren't hit by the SARS outbreak at all, we didn't put in place the measures that we needed to combat coronavirus effectively. So there are 3 ways to stop an outbreak: is to prevent the outbreak from happening by basically just not getting infected by animals. The second is for finding a vaccine, which is what we're trying to do now with coronavirus. Because we cannot prevent the outbreak from happening already, we now are trying to find a way to cure it. So that comes with the vaccine. And then the third technique that we need to use is social distancing or containing the outbreak. If you know what happened in China, the Chinese government was pretty good at stopping their outbreaks by just shutting down all public transport, which is something that we can't necessarily do because we live in a democracy. But but basically, by containing and stopping transport of people in and out of Wuhan, they were able to contain the virus very quickly and very effectively. But the way that we're handling here by social distancing is also very good. And the stay-at-home order is also very good because it helps us to have less interaction and stop the spread of our virus outbreaks. So why is China susceptible to coronavirus? Coronaviruses, which is both the SARS virus and the coronavirus that we're facing today, COVID-19, are both coronaviruses. They're both the same nature, and they're both from the same umbrella. So China has an unregulated wild animal market created in the 1970s when the food production stopped being state-owned. And so they opened these markets, and since animals like bats and sea snakes often carry these viruses that can be transmitted to humans. And both of the outbreaks were caused by these wild animals infecting the people. So China's infrastructure also spreads disease because they have a bunch of state-owned transportation systems that are often very cheap, allowing for the disease to spread very fast when it breaks out. So having mass public transport can also hurt a country because you're having a lot of people in close contact. So what is our solution? So obviously we're trying to find a vaccine, but quarantine right now is our best option for containing coronavirus. We're already past the stage of trying to prevent it from becoming an outbreak. Okay. So yeah, I mean, in a sense, trying to prevent the outbreak from happening at all is a good answer. I probably should have looked at that before. But in our case now, since this was made a little bit earlier, in our case now, B and C would probably be the best, our best go at it. So if we don't have to do this activity because it's like bingo and it requires a whole different site. But basically, it talks about all the different viruses and just basically important stuff. So right here, I'm going to hand it off to Sam, who is going to be, taking over. Okay. Sam is not responding right now, so wait, am I just not hearing him?
No, we can't hear you.
Can anyone hear Sam? I cannot hear him. Please respond in the chat.
Wait, Mason, can you hear me?
Okay. You know what? I'll just continue. No. Okay. So, oh, okay. So I'll just continue while he tries to figure that out. Sorry, you're going to have to stick with me for a little bit longer. Okay, so let's get into introduction to virus construction, or basically how viruses are built. And this presentation was created by my good friends Aditya and Vincent, who are both pretty experienced in the biology field. So basically, we already talked about what is virology, but a good recap of what we missed in the recording was how virology is the study of all aspects of viruses, including how they spread to what they're made of. So basically, not only the nature of their function, but also the reason why they're so effective, and the reason why it's debated on whether they're live or dead, and why they're classified as a separate organism or separate thing, really, than bacteria and fungi. So let's look at what a virus is. So basically, the major features of a virus include the membrane, and then the glycoproteins, or like when you take a look at a diagram of a virus. Let's see if we have one here. Yeah. So we have these proteins on the outside, and so that's their glycoproteins. And then your matrix proteins, which makes part of that rigidity aspect to the virus. And then, of course, the most important part, the RNA, or the genetic code, which that virus spits into a cell, causing it to create more viruses. So let's look at our virus. So if we take a look at a 2D model, as you can see here, we have our proteins on the outside. And then if you look at here, we have this membrane bilayer, and then the matrix protein right here, which is what we're talking about, the rigidity of it. It's not the same thing as a cell wall, but it kind of helps with the shape of the virus. And then here we have our RNA polymerase, which we talked about polymerase last week, so you guys should know that. Can anybody tell me in the chat what polymerase is? Anybody, give it a go. Yeah, so no one, very funny name, but basically, yeah, you're right. It's an enzyme that helps with the formation of DNA or RNA. Okay, so let's continue. So, so this is a 3D model, which kind of gives you a better look at our viruses. And so as you can see here, here's our RNA inside here. And then we have our matrix shell and the proteins on the outside. Yeah, pretty cool, right? Okay. And this is a 360 model, which kind of looks a little bit funny, but I guess it gives us a good look at, I don't know, this animation. but yeah, so basically it gives us a good look at a model of a virus. Okay, so there are multiple different types of viruses that we can talk about, and this includes different shape viruses. Even though we took a good look at a standard virus shape, there's many different forms or shapes a virus can come in. Not only that spherical or elliptical sort of shape, but there also is polyhedral, helical, complex, and enveloped. And these are all the different types of viruses. Polyhedrals are composed of polyhedral protein shells. Helical are composed of cylindrical capsid with helical symmetry, and complex are composed of many proteins that work together to carry viral functions, and enveloped are surrounded by a lipid membrane that includes glycoproteins.
Hello?
So when we talk about a polyhedral virus, it obviously comes from the shape. And you guys know what a polyhedral shape thing is, right? So basically, like, it has like these triangular sort of sides, and but yeah, and it creates this sort of big thing. Yeah, great explanation. But basically, they consist of nucleic acid surrounded by a polyhedral protein shell, and are usually formed of an isohedron. And this is a non-enveloped virus, and two examples of this are adenovirus and the poliomyelitis or polio. Okay, so when we talk about helical viruses, they consist of nucleic acid surrounded by cylindrical capsids with helical symmetry, and they're not enveloped, just like the polyhedral viruses. And an example of this is the Paco mosaic virus, which is a callback to our previous slideshow. So an enveloped virus is what those two aren't, and they consist of nucleic acid surrounded by either helical or polyhedral core and outer lipid membrane. So when we talk about that, if you look here, so basically it's this. Nobody can hear you. Wait, chat.
My audio works again.
Oh, okay.
Okay. Should I take over now?
Okay, bye. Sorry guys, I can't hear you, so I'm going to log out.
Is this better?
Whoops. Sorry about that. I can hear you now. Yeah, Sam, I can hear you, so go ahead. Okay, he left. So I guess I'm going to continue. Sorry for the interruption. So let's continue. Okay. So an enveloped virus is a virus that consists of nucleic acid surrounded by either helical or polyhedral core and an outer lipid membrane. And examples of these are the dengue virus, influenza virus, and the measles virus. And it's a more common type, and it's a more sophisticated type. Okay, let's continue and try to get back on track. So when we're talking about a complex virus, we're also talking about bacteriophages, retroviruses. They also come in this shape. And basically they're a combination of an isohedral and a helical shape, and may have a complex outer wall or head-tail morphology. And this is usually the thing that you think about when we're talking about viruses, because it's not just a blob, and it's more of like an actual thing that injects stuff into cells, and it's more sophisticated. That's why it's called the complex virus. Yeah, good job. A microphage. It looks like a bacteriophage, if that's what you're referring to. And the reason why it looks like so the reason why it looks like a bacteriophage is because bacteriophages are bacteriophages are a type of virus, and it's a virus that infects bacteria. And if you guys don't know, bacteriophages are currently on kind of the edge of medical treatment for diseases involving bacteria. Previously, we've been sticking closely to antibiotics and trying to develop more and more potent antibiotics in order to combat bacterial problems. But what we've noticed is that these antibiotics have become less and less potent. Bacteriophage, it's because they're a type of virus. But yeah, So bacteriophages are really, really important right now, and it's not only are viruses important just because of coronavirus, but bacteriophages are especially important because a lot of research has gone into bacteriophages and how they can cure diseases, like they have done in Europe, and they're actually commonly used in Europe to treat the diseases more effectively now than antibiotics, because antibiotics have become less and less effective because of their overuse. So let's get back into this. So to review our structures of viruses, we have the main structures, which are the lipid. Oh, hi, we can hear you.
Okay, great. I don't know what happened to my audio, so I guess I'll pick up with the next mini presentation.
Okay, let me finish up real quick. So to review real quick, we have the lipid membrane, the glycoproteins, the matrix proteins, and the RNA. And there's 4 main types of viruses, which include the polyhedral, the helical, enveloped, and the complex virus. Okay, I'll hand it to you.
Yeah, sorry my audio wasn't working. I just got it working right now. Okay, anyway, so this is going to be about the epidemiology and the applications. And epidemiology refers to the study of disease in a population. So we're not looking at one person or the effects of the disease in one specific person, but rather we're looking at the effects of the disease in an entire population. So we're focusing not on an individual, but on among a entire group of people. Okay, so yeah, obviously, as you guys have learned, the scientific method is the OG thing. It's everywhere in science. It's literally it's one of the most important things. Curiosity and the scientific method. If you have the scientific method, you can pretty much have a valid way of making or conducting an experiment, and the same thing applies here. So with this, the scientific method, as you guys know, you've learned this many times in science classes. The first thing is to obtain background information, and in with relation to the epidemiology, what that entails is preparing for field work or going out into the actual town where the virus is occurring. Like say you go out into Wuhan and you collect samples or something like that. That's I guess a example. And you can also establish the existence of the outbreak here. Establish the existence of the outbreak. Uh, Consider severity, potential for spread, public concern, and availability of resources, and verify the diagnosis of like multiple people, and you can do contact tracing. So that's like part of the background information that you obtain about the specific outbreak. The next step as part of the scientific method would be to define the problem. So as part of that, you would define and identify the cases, and you describe and orient the data. So demographics are very important in epidemiology, as you don't want to compare an old person to say a young person. You want to have everything categorized in terms of their age, gender, etc. So there are a lot of factors in which you have to list everything out. The next thing, to formulate a hypothesis. In this case, the hypothesis is in the form you have to include the agent, host, environment. Those are the 3 things in the triad, and that allows you to see the chain of transmission. The next step in the scientific method, developing the study to test the hypothesis, collecting data and observations, and evaluating the results. That's basically, as you can see, you just conduct the actual study. And it's very important to have a control group in order to compare your results from your experimental group against some background level, so you can compare it against something. You can't have one group. You can't just have one experimental group and not have a control group and have it be a valid experiment. And finally, the last step or few steps, determine if the hypothesis is true / modify it, formulate conclusions on it, and report results. So again, this is pretty much what you get out of the study, and you based on the results, you can refine your hypothesis to better fit the data and carry out additional studies to make your data stronger. So yeah, and then you can, based on your data, you can also implement control and prevention measures, and you communicate your findings to other scientists so that they can verify your data and make it stronger. So essentially, these are the 10 steps required for studying an outbreak or studying an outbreak of disease anywhere. as we covered before in the outbreak investigation steps. So this is just a little bit more specific for the first step, preparing for field work. So what that specifically entails is the people have to do research, a little bit of prior research before actually conducting research in a study. They have to research about the disease and then gather all the necessary supplies and equipment to conduct their investigation. They have to make arrangements with the local officials in that particular city in order to announce their visit and what they're going to do. That all takes time, and that is all necessary part of the investigation. And again, they have to follow the protocol. They can't just do things willy-nilly as people often do in movies. Okay, so the second step, establishing the existence of the outbreak. So what epidemiologists often do, they go to the health department and obtain hospital records, death records, physician records, and they obtain surveys from doctors to determine the expected number of cases in a certain area in a given time, as that's really important to confirm the existence of the outbreak and the actual number of cases. They have to actually take into account other factors as well, as these may skew the data. So if there's not good reporting of the data, that'll affect it. Seasonal fluctuations, they have to take that into account, and population changes like immigration, emigration. They have to take all of that into account. Okay, the third step, verifying the diagnosis. So it's a little bit self-explanatory, but we'll go over what it exactly means. So as the name implies, verifying the diagnosis, you have to verify the procedures used to diagnose the problem and check the methods used for identifying infectious and toxic chemical agents. You have to have a consistent way to identify the disease. You can't just have everyone use a different method, and so that way you can obtain accurate data. Though for verifying the diagnosis, it's also important to make sure that there's no, or you want to try to reduce the experimental error or lab error, and make sure that the increase in the number of cases is not due to random chance or something erroneous in your methods. And finally, the commonality. You want to interview multiple people who became ill to gain insight concerning the possible causes or sources of the disease. As if you do it to one person again, you're not going to get good data. In order to get good data, you want a large number of people that you want to interview, and your population sample has to be pretty big if you want accurate data. Okay, for identifying and defining and identifying cases. Okay, so epidemiologists actually have to define what the case actually is. So they use 4 standard criteria for determining who has the disease or condition. So these are the 4 criteria. Number 1, clinical information about the disease or condition. It's like, for example, what they have a dry cough, or everyone who has this disease has a dry cough, or rather, a majority of the people who have the disease have this symptom. Second, characteristics of the affected people. Third, location or place. You want to have it localized to one area or where the outbreak occurred. And lastly, you want to have a time sequence during which the outbreak condition occurred. Okay, so this is an example for influenza. The case definition for influenza. So a case of influenza-like illness or influenza is defined using the case definition as a person with fever of 100 degrees or greater orally. So they do specify where they take their temperature, and cough during the influenza season. So there's the time and there's the symptom, and they have a very specific kind of case definition, which is necessary. And then a person with laboratory confirmed influenza is also considered a case, even if the person does not have cough and fever. So as always, there are always exceptions, but these are always explicitly defined so they can fit within the case definition. Okay, identifying cases. So the identification of specific cases, the kind and number, that has to do with they have to be confirmed. They're basically categorized into 3 kind of categories. Confirmed, probable, and possible. these are the three types of cases. So the confirmed case, it means you have the diagnosis and you fit the case definition, plus you have lab verification. This is kind of like the highest level. That means you have 100%, almost basically 100% have the disease. Probable means many factors point to the diagnosis, but you may not have lab verification. That doesn't mean you don't have the disease, but it doesn't mean you 100% do have the disease. But it's just likely that you do have the disease. And possible is the weakest out of the three. That's just saying some factors point to the diagnosis, but you may not necessarily have a strong case for having the disease. And yeah. Okay, so line listing. So line listing is basically a chart of specific cases, including information about each case. So that's the kind of thing with the demographics that's very important. This is kind of just organizes the information visually, so it's easy to look at, and it's not just huge massive data. So for identifying information, you use an ID or case number. There's a left column which has all the names or initials. And then in the right columns to the right of that, there you have clinical information with the diagnosis, symptoms, lab results, hospitals, or even death if the person died or not. You also have descriptive columns with the time, date and time of onset, plus data report, descriptive person, and with the age, sex, occupation, or other characteristics. You also have the place, which is the street, city, or county, or a specific site where they either contacted or acquired the virus. Yeah. And then you have risk factors and possible causes specific to a situation and outbreak setting. Okay, so this is an example of the line listing. As you can see, the leftmost column right next to the ID is the ID column, and then to the right of that, you can see the different columns with the different demographic information. Okay, so this is the line listing for the coronavirus. As you can see, it's organized by country, and you can see the case numbers, the new number of cases each day, the total number of deaths, the new deaths per day, the total number recovered from the virus, active cases, and so on and so on. You can see that they do include quite a lot of information, and you do want to be as specific as possible. Yes, it is an old list, but these things are constantly updating every day. But that was the list. That was the line listing at that time when we put that in. Okay, so this is the time, place, and person triad, which describes the form. Yeah, this describes the forms of that. So the time, basically, it's a histogram showing the course of the disease or outbreak to identify the source of exposure. The epicurve, it begins early, and they update it quite often. They have the place, which is the geographic extent, plus the spot map of cases to identify groups specific to a location or environmental factors. They have the person part of the triad, which identifies the affected population by the type of person or by exposures as age, sex, high risk exposure as with AIDS. So this is a sample epicurve, and just make sure that you see that a single point or source will only have a peak or a plateau. A plateau will show a continuous common source, and several uniform peaks will indicate a propagated outbreak spread from person to person. So based on the shape of the curve, you can see kind of the outbreak it is. So these are the main types of epicurves. As we saw in the other epicurve, that you can see that you can see how this is categorized. So there's point source. That's when epidemics have persons that are exposed to the same or have the same exposure over a limited, well-defined period of time. The shape of the curve commonly rises rapidly and contains a definite peak followed by a gradual decline. So this is a sharp peak, and there's nothing ambiguous to it. The continuous common sources are when exposure to the source is prolonged over an extended period of time and may occur over more than one incubation period. So the downslope for this curve may be very sharp if the common source is removed, or gradual if the outbreak is allowed to exhaust. So it's very important to gather information about that kind of data. Okay. Okay, so these are the other two types of epicurves. Propagated, or when epidemics occur when a case of disease serves later as a source of infection for subsequent cases, and those subsequent cases in turn serve as sources for later cases, kind of like what we're seeing right now. The shape of this curve usually contains a series of successively larger peaks, reflective of the increasing number of cases caused by person-to-person contact, until the pool of those susceptible is exhausted or control measures are implemented. An example of a control measure being social distancing. That's the whole point of what we're trying to do, to lower the peak of the curve and not have it as high as possible. And finally, the last one being the intermittent common source. And in this, the pattern reflects the intermittent nature of the exposure, and there's irregular intervals for the curve, or the peak of the curve. So yeah, here you can see what a point source looks like. There's a well-defined peak, and just yeah, it's very recognizable. This is the continuous common source. You can see it's a little bit of gradual. It's a little bit gradual over a period of time, and then once the exposure is removed, you can either be gradual again, or it can be sharp if the exposure is abruptly removed or it's slowly removed. Okay, so this is the next one, the propagated or progressive source. So this comes in little spurts with multiple peaks, well-defined peaks, and this is person-to-person contact. And finally, this is the intermittent common source. This is how that looks like. Okay, so this is the coronavirus epicurve, as we can see. Again, yeah, this is not the most up to date, but this does reflect the overall trend. So you can see the increase, the sudden increase highlighted in red with the total case under the total cases. Okay, so what type of epicurve do you think the coronavirus follows? Just answer in the chat or out loud. Okay, so any more answers, or is it just will? Okay, yeah, it's point source. So you can see there's like a well-defined. Okay, so you can see there's a well-defined curve for the coronavirus epicurve, and therefore you can see that it's a point curve and not any other type of curve.
Okay, so I'll get into the types of descriptive studies. So there's multiple types of descriptive studies, as you can tell by the title, for sure. So what a descriptive study is the study of the distribution of a problem by cases or outcome. So it basically describes the frequency and population, the exposure, the time pattern, or the environmental factor. And basically, we get into case reports and case series. And what a case report is a detailed report of a single patient from one or more doctors. So basically, when we're talking about case reports, we're talking about individual cases. And you often see this not only in when we're talking about viruses, but also when you talk about psychology and just science in general, when you specifically study a single patient, that is called a case study. When we get into a case series, it's very similar to a case report or a case study, but it's just more of those people, and probably a little bit less in depth on than the single person case. When we talk about correlative studies, is the complete opposite of a case study, in the sense that we don't go very in depth on one person, but we get a average of a ton of people. So basically, we get general characteristics, and in this case, with diseases. And we correlate different factors with one another and see if there's any correlation between certain factors and the outcome of diseases. A time series analysis correlates within one population, but through different points in time. So we follow just one group of people and go into throughout time. And ecological ecologic relations finds correlation relative to specific ecological factors such as diet, home, work site, or travel location. So basically, it compares groups of people based on their activities. And a cross-sectional is basically when you take a person and you see how they are affected throughout time, and you check up on them every so often. So when we develop our hypothesis, we have to look at different factors. Agent host environment is a big one. It's often considered, it's often referred to as like a three-pronged sort of idea. And it's the agent capable of causing a disease and its source, host, or persons, the susceptible to agent plus the environment allowing them to get together. So these three things can help us form our hypothesis if we know all three of them. And we can determine it based on the different groups, including viruses, bacteria, protozoa, fungi, and animals. And right now I know we're specifically talking about viruses in this case, but this is just in general for all epidemiology. And our hypothesis must be testable. So remember when we were talking about hypothesis in sciences, you have to ask a question or give a prediction, but that prediction we must be able to find it by the end of our study. So we don't know it yet. We don't know the outcome of it yet, but through the study we're able to find the answer or find some sort of conclusion. And to develop a good hypothesis, it's good to get a lot of background in, just so you ask the right questions and look for the correct answers. Yeah, and your hypothesis should get updated as you receive new information. Okay, so when we evaluate hypotheses, we compare with the established fact that these are used when evidence is strong and clear cut. So remember, hypothesis isn't just some sort of guess out of the blue, but it's more of like, okay, we know this and this. So what is going on here? Could this could this outbreak be caused by, let's say, a virus? That might be our hypothesis, or something like that. And then you can also evaluate hypotheses through observational studies, which is kind of more a standoffish approach to evaluating your hypothesis, because you're just studying things and instead of interacting with them like you do in experiments, sort of. Okay, I'm just gonna admit this person right here. And then we can go into cohort studies, which is based upon exposure status and whether they not have the illness, and how that is related to exposure. So that you're basically comparing direct exposure to if they have the illness or not. And a case control study works backward from effect or illness to suspected cause. So instead of cohort, which we're going forward, we're working backwards with case control. And to verify your hypothesis, you must have actual lab work or have actual tests done. And you can't just use hypothetical data or like cohort data. So when we talk about cohort studies, both groups have a known exposure and are checked for future outcomes or illnesses. When we're talking about retrospective cohort studies, we talk about when it starts at the exposure and in the past and moves forward to the outcome in the present. So we know these people have been infected with the virus, and now we're looking at them to see if they have the disease or they've been exposed in this location. So we're looking to see if they have the disease. And then when we're talking about prospective, we now infect them and see in a while to see if they have the disease or not. So when we talk about cohort studies, we always talk about the 2x2 table. This is a big thing. And when you talk about diseases and everything, if you took Science Olympiad disease detectives, you have seen this a million times. I mean, this is a big thing. So when we, so it basically compares here whether they were exposed or not and whether they have the disease or not. And we do a little bit of math here and we see the ratios of if they have the disease versus whether they have exposed, or yes, no exposed versus if they have the disease or not. So basically, this is a game changer. And then incidence versus prevalence, basically, this is our calculations when we look at our cohort table. So incidence rate is the amount of new cases in a population. And you do a little bit of math from our data table, and we can define the incidence rate. And the prevalence rate is the amount of new and pre-existing cases in the pre-existing population or at risk. So prevalence is basically everything, and incidence is new. So that's the difference between the different case rates. So when we're talking about morbidity versus mortality, morbidity is the same as prevalence rate when we're talking about how many people have been infected. And mortality rate is very different. It's talking about how many people are dead. When we're talking about coronavirus, it does have a high death percentage, but most people that do get the virus are not necessarily dead. They'll get very sick, but they're not necessarily going to die. So that means that your mortality rate is going to be lower than your morbidity rate. But while they sound similar, they're completely different. And then crude mortality rate is basically taking the mortality rate by various factors, and taking the total death rate and separating them into groups based on their different factors. Okay, so when we're calculating attack rate and relative risk rate, we have to look at our 2x2 cohort table. So when we're talking about the attack rate, is the rate that a group experienced an outcome or an illness, the number of sick people divided by the total in that group. So if we do our little bit of math here, we can, the exposed is A here divided by A plus B in the total range. And that is going to give us an 80% exposed outcome when we're talking about how many people were exposed and whether they do or do not have the virus. And when we're talking about unexposed rates, we take here C, 50, and we divide it by the total 220, and we find the prevalence rate of those unexposed people. And when we calculate the entire relative risk, we take the exposed rate divided by the unexposed rate, and we find that number. And basically, relative risk, you can measure it and see how dangerous the disease is based on its relative risk number. So interpreting results of cohort studies, relative risk estimates that the extent of the association between an exposure and disease, and basically allows us to take a look at how many people have been exposed and how many people actually got the disease and compare the two things. And then when we talk about when the relative risk is greater than one, that causes, that indicates a positive association or high risk. And when the relative risk equals one, that means that the exposed group is equal to the incidence rates in the unexposed group. Therefore, there's not enough evidence to provide association, and therefore being exposed in that manner is not probably going to be the factor why people are getting sick. And this relative risk is not expressed in negative numbers because we're dividing things that are positive. Okay, let's move on. So case control illness. Basically, we're working backwards from vector of illness to suspect the cause. And that basically works the way that you expect it to. But we do have the odds ratio, which is another sort of math thing to help us determine the susceptibility of this disease and to evaluate the agents and vehicles of transmission or how those people get diseases. So when we look at our sample case control table here, this is another example. We don't really need to go through it, but basically, you know, we take the same look at exposure factor versus actual patients and controls. And as you can see here, these patients here, whether they did eat or did not eat, and see, okay, let me rephrase that. So we have case patients and control patients. And basically, case patients are the people that are diagnosed, and the people that are the control patients are undiagnosed, or the people that do not have it. And then the did eat or the did not eat is our exposure factor. Remember, exposed versus unexposed. And we can calculate from there our different rates. So when we look at the odds ratio, however, Basically, we're talking about odds of exposure in cases divided by the odds of exposure in controls. So basically, we're looking at how likely are they going to be exposed if they don't have the disease, or versus how likely they're going to be exposed if they do have the disease. And so basically, when we calculate the odds ratio and it comes out to 5.8 in this scenario, that means that people who ate at restaurant A were 5.8 times more likely to develop hepatitis A than the people who did not eat there, because the control patients did not get the disease. Does that make sense? Okay, I don't care if it makes sense. We're going to continue. Right. Yeah. Let's redefine the hypothesis and conduct additional studies. So when there's no confirmation of the hypothesis, you have to make a new hypothesis. So basically now you'd be like, oh, that failed, so let's try again. And you try to look for a new vehicle or new method of transportation. And you might have to tweak when you find evidence that sort of supports your hypothesis, you might have to tweak your hypothesis and redefine it, and then take another look at the data or collect more data to see if that supports that new hypothesis. And then, you have to do those environmental laboratory studies if you found evidence that kind of supports your hypothesis. And to find confirmation for your hypothesis, you have to work in laboratory settings. Okay. Let's get this. So when we're talking, do you want to take over, Sam?
Yeah, sure, I'll take over. Okay, so again, this is the chain of transmission that we were talking about. So here, let me just pull it. Okay, so with the chain of transmission, you can see the infectious agent, the reservoirs are where the agent kind of, or the pathogen kind of stays, the portals of exit where it leaves, the modes of transmission, the portals of entry, and the susceptible hosts. So that kind of defines what each of the things are, and this is kind of like how the disease is spread from point A to the end or to the host. Okay, so with disease and disease transmission, the agent is, as we talked about, an agent. Agent is a microbial organism with the ability to cause disease. Reservoir is a place where agents can thrive and reproduce. A portal of exit is a place providing a way for an agent to leave the reservoir. So portals of exit tend to be like pretty well defined. What serve as portals of exit are often not terribly surprising, but like once something is known how and where a pathogen replicates and enters a new host, we can utilize that to maybe stop the chain of transmission in that one place and thus break the cycle of infection and stop new infections. Respiratory infections tend to utilize the mouth and nose as portals of exit. Gastrointestinal diseases tend to exit in feces or saliva. Sexually transmitted diseases tend to have portals of exit at the urethra or genital region. And blood-borne diseases tend to exit via insects like arthropods, needles, bleeding, or hypodermic syringes. Okay.
Okay. I'm afraid I'm going to interrupt you there because we are out of time.
Yeah.
Unfortunately. Sorry. We didn't realize that the time's almost up. So yeah, thanks for coming. Just to recap real quick. Viruses. Let's go back. Okay. If we're going to recap real quick, virology is a study of viruses. I know that this got cut from the beginning, but virology is a study of viruses, and we can use the information that we acquire from studies to develop hypotheses and find ways to cure or provide vaccines for these different viruses. Okay, thanks for coming, guys. That wraps it up for the session. I hope you learned a lot about virology. Yeah, I can send you the slides if you want. Just put in your email. Yeah, that's basically it. Thanks for coming.
Thanks for coming.
Yeah, no problem. Can you post the link in Docs? Sure, I can post the link in Docs for sure. Yeah. Yeah, no problem. Thanks for coming. See you guys next week. Yeah, no problem.
Bye.
All right, bye guys. Yeah, bye. Thank you. Bye. Oh, wait, who's recording? Bye. Yeah, thanks for coming. Okay, I'm gonna start recording now.